Hello, welcome to Biology Videos with Mr. Lomas. I'm going to be doing an introduction. This is designed for Year 11 students who are looking to do either A-level biology next year or applied science. I'm going to be basing these lessons on the OCR spec, and I'll be quoting the codes from the Caboodle textbook. So if you study at TDA, this will be the code of the books that you will get. This is still applicable for current Year 12s, current Year 13s. It's going over the exact same stuff. It's going to be more of a steady introduction, more step by step rather than kind of firing too much. And the idea is this should get you ready for the beginning of September and you can kind of start on it now. Okay, so we're going to be looking at microscopy the best we can without doing any practical. So the theory around the microscopy, and then going into some OCR requirements you have to be able to do, such as biological drawings. By the end of the lesson, you should be able to look at light microscopes in detail, and we'll come on to electron microscopes in more next lesson. Okay, so the microscopes themselves. There's a long development of microscopes that have started almost from the Roman times, in which they started to use glass, and there's some reports from previous to that in the 13th century. One of the first kind of discoveries of what we call the compound microscope is from someone called Galileo. Now, you may have heard him before. In 1609 he produced the first compound microscope, and that takes us to what from here to what we're at now. This should be reasonably familiar to you in terms of the light microscope, still a compound microscope that we use in schools. So we're going to look at the development of this microscope now. So these are some of the key dates that I've pulled out from you. Again, these are OCR specific and I'll highlight them. So one of the first key dates is in 1665 from Robert Hooke, where this was a drawing by himself of his own microscope, where he discovered, by looking at some plants, he discovered some cork. He found these round little dots on this cork, and he named them cells similar to monk cells in monasteries. So he coined them the term cells, which is one of the very first time this word cells was used. And that takes us then to Anton van Leeuwenhoek, or Leeuwenhoek, it depends who you ask. And he was one of the very first people to discover living organisms other than plants and animals, and he called them animalcules. Now, if you can imagine being in a situation where you're the very first person to ever see some new microorganisms, these tiny, and these are some of the drawings that he essentially did, and these are retouched versions of those. So these two guys are two of the very first guys to look and use microscopes in the 1600s. And now this takes us on to some more recently, and it will have accelerated since then, but some other sort of key points going a little bit further. So one of the key ones is Robert Brown in 1833, was an English botanist. He discovered the nucleus. We also had a variety of different scientists, so Ramac and Virchow, and they understood cell division, so what you know from GC is mitosis. They were some of the first people to document on that. And then we've got kind of cell theory, the universal acceptance that everything's made out of cells, which came around and was more well evidenced around 1937. And there's lots of other ones. So some key ones for us to learn is Robert Hooke, Leeuwenhoek, and the universal cell theory. Now that takes us to the compound microscope. So I'm going to go through very quickly what each part of the microscope does, and I'll give you a more detailed version. So the microscopes that we use, we have this turret, this rotating nose piece, which is used to spin, and then you've got an objective lens in there. So this is one of the lenses, and then you have a second lens in the eyepiece. So in each of these microscopes you have two lenses, and all you're doing when you're looking through is you're seeing something on the stage, which is held there by these stage clips. You're seeing some light which is passing from behind, that's passing through an image. That will go through the first lens to magnify it, then the second lens that magnifies it again, so it will make an image appear much larger than it is in real life. Now, the kind of using these microscopes, at A-level they're slightly different. In the eyepiece there's a graticule, we'll talk about them more in later lessons, but there's also, we tend to have this coarse and fine adjustment, and all these adjustments do is they move the stage up and down, and that means that we are moving the specimen that we are looking at on the stage into the focal plane. So when we look through the lens, the first lens and the second lens, there's a point in space and time. Now it might be where my mouse cursor is now, which means the stage will be too high, and that is why it looks blurry. So if we then move the stage down, we'll then focus on this exact place in space for us to focus on. We also have this diaphragm underneath, or referred to as a condenser, which is like a little clip here, and you turn it, and this will condense or focus the light into different parts when you look under the microscope, and it essentially allows it to brighten up or darken a specimen by focusing light in a particular area. Okay, so moving on. I just want to give you a bit more detail in terms of the way the lens works. The key point with this combination of these two lenses and these distances of the lens is this reduces something referred to as chromatic aberration. Now, this is something you need to learn. Now, every time light is used, and we use white light, which is a mixture of wavelengths, and it passes through a lens, it will refract, but it also will start to disperse. It will spread apart because the different wavelengths bend at different amounts, or change direction rather than bend. Now, blue light, and the way that I always remember this was blue bends best. So blue light will refract the most, and red light the least, because they're opposite ends of the electromagnetic spectrum. So that essentially means that light's going to spread out, a bit like the rainbow. It's going to spread out, and this causes a blur. Now, when we're viewing something, and this is my focal plane, this is where I'm focusing the light onto. This could be the specimen that we're looking at, and this could be the slide. That means the light is going to be spread out, and it's going to make a blurry image. So the idea of the design of these microscopes, the way that we overcome that is the positioning and the way that the different lenses are used. That's attempting to reduce the amount of chromatic aberration. So that's the spreading of light, and the failure of the lens to focus all the wavelengths of light on one particular point. So that's what chromatic aberration is. Another couple of key terms which are important is magnification, so how much bigger a sample appears than it is in real life, and you should have remembered from GCSE the I-am equation, so it's exactly the same as here, so the image, actual, and magnification. We're going to go through this in more lessons. And then that takes us to resolution, which we don't look at in very much detail in GCSE, so I'm going to try and explain it now. So it's the ability to see 2 points, often referred to as separate entities. Okay, now magnification and resolution are different things, and I'll try and explain the difference. Resolution is the ability to see 2 images or 2 places as separate points. So on this example that I've made here, you've got perhaps 2 cells here, and you're looking through the microscope, and the resolution is lower, so therefore you can't see it as well. So you've got this blurring of these 2 images overlapping. As we increase the resolution, now this is not increasing the magnification, just the resolution, it allows us to detect the differences between the 2 cells as separate entities. So a very rough way to say it is it's how clear the image is the resolution, but you should describe it as the ability to distinguish between 2 points as separate entities. Now, the resolution of light microscopes are about 200 nanometers, and electron microscopes are much better. We'll get to those more in another lesson. Now, just some prefixes and symbols that you're going to come up in A-level biology quite a lot, kind of starting normally from millimetre. My number one piece of advice is always work in millimetres. Try to refuse to work in centimetres unless an exam question specifically asks, and they very rarely do, because that allows you then to change into the prefixes between micro and nanometres quite easily, and you know it's 3 more zeros in short. Take note of the symbols, so you've got this micrometre symbol, nanometre, again we'll do more in later lessons, and sometimes we even look at picometres as well. Okay, so to sort of summarise light microscopy, it's cheaper than electron microscopes, and we'll get to those next time. They're small and portable. Good things about them is you can view cells that are living or dead, and there's even really cool light microscopes that have even been attached to animals' brains, and you can view brains whilst people are alive, and various other things that are being developed at the moment. They have a magnification of up to 2000. Now that means you might get an exam question where it says 1500, which is quite a common exam question from older OCR questions. That doesn't mean it's incorrect, because it's a magnification of up to 2000. Now they're getting slightly better, but the reason why they're limited to around the 2000 is because of this wavelength of light. The wavelength of light limits the ability to magnify images further, and we'll look at magnification more next lesson. Resolution is 200 nanometers. Another advantage of light microscopy is that it allows us to stain images in colour, and allows us to distinguish between 2 different organelles in a cell and see a contrast between them. Okay, so now I'm going to do a quick quiz with you. So get a piece of paper, have a go at these questions. Okay, so you can just go back in the video and you can look at the definitions for resolution, chromatic aberration, magnification. Name 3 contributions. Again, you can go back to the previous slide and have a look at 3 contributions. We're now going to do some multiple choice questions. So which one of these 5 is correct? Again, as I go through these questions, just pause the video and then play it when I come to the answer. Okay, so the one that's correct is magnification is the size of an object image in relation to the size of the original object. So it's that relationship between the two. How much bigger does it appear? Okay, next question. Okay, so resolution is how much of an object's detail can be viewed. Now this is a very crude, a very basic way to explain it. I would advise always saying resolution is the ability to view 2 objects as separate entities, but this is kind of a generalistic detail can be viewed. And finally, a high resolution, what can it do? Well, it can tell us 2 objects that are very close together apart. Next question. Okay, so images using a microscope with a high magnification and low resolution are often blurry. So resolution is the ability to see an image clearly. Next question. Okay, so optical microscopes use visible light and they use glass lenses to do that. And final question. So we can see, and I'll come to this more in a second, living organisms such as bacteria with the light microscope. Visible light is the limiting factor, and we can't see ribosome. You should remember that from your GCSE. So let's look at some images. So I'm going to start off with some images. So these are examples of bacteria. These are human cheek cells that go in there. We can look at goblet cells and ciliated epithelial cells. You should remember these from your GCSE. These are the cells that flick mucus out of the throat. The goblet cells are the ones that produce the mucus. We can look at cartilage cells within the body. So these are the cells that you might find in the trachea. We can look at alveoli, we'll go to more detail in a second, and bronchioles and the structure of that, and we can see this increased surface area here with the folding. And we can look at squamous epithelial cells as well, see that they're very thin for increased gas diffusion. And what this allows us to do is look at all different parts of the human body and then learn from them. Now this kind of image sort of shows here on a logarithmic scale the ability to look at objects at different size via different microscopes. So for instance, we can see an optical microscope can get down here to just about halfway between a micrometer and 100 nanometers. And this kind of is around about here where you'll be viewing cells if you study at A-level or applied science. And for instance, red blood cells are quite small under the microscopes we use at 400 magnification, and they're about 7.5 micrometers in size. So we'll be looking at things kind of around here. And you can see electron microscopes are much more powerful, which we'll come to another lesson. So you can see here, we've got a cool video here of an amoeba that's eating a paramecium. So this is able to be viewed under a light microscope, and you can see that these pseudopods of the amoeba, so they're the bits that stick out, and they're kind of these branches of the cell membrane. They will surround, and then they will engulf by phagocytosis, as you've learnt for white blood cells at GCSE. And then you can see this little paramecium squirming around on the inside, because they're going to be broken down. So there's going to be these enzymes inside here breaking down these paramecium and then killing them off and essentially having them for lunch. So we've got an image here of Clostridium difficile bacteria, which is an antibiotic resistant bacteria that we can see. And when you look down the microscope for the first time, you're going to see this little graticule along here, and we use this graticule once we've calibrated the microscope to measure. So in this case I can spin my graticule or I can measure it, and we'll learn more about this when you do the practical side of it. I've also got this example here of some mitochondrium and some kind of other organelle in there. We've got images of chloroplasts themselves, these little green chloroplasts that look pretty cool under the microscope. So this is a kidney cell that we can see here, and then we could measure the size of the nucleus, the nucleolus within inside the kidney cell. And then we've got some images of onion skin cells, so you've likely have seen this kind of thing before. You can kind of see this brick style shape that you'll have seen before, and you'll likely do at some point if you do A-level. And then finally we've got some ciliates that are about 100 micrometers in length that we can measure. And there's lots of things that we can measure with microscopes, and here's just some examples. So the application of what we looked at so far is you'll likely have to do in your exam, as well as your practical side of the course, is some biological drawings. And here is, and I'll try and link it the best I can, the biological drawings handbook that basically gives you every piece of information in detail. It's roughly about 20 pages long, I think, and I've cut bits out for you. So when we look at cells under the microscope, for instance, here's a group of cells in the pancreas that we're looking at, and this is a lobe, so this is a tissue image here. We will have to do images of that, and this is an example of a good biological drawing to the right. And you can see the roughly similar proportional size. Okay, it's not quite exact, but it's quite clear that this person has drawn this this set of tissue here. We've got another example here in a pancreatic duct where you've got some cells. Now this has got a higher magnification, so we're drawing just some cells, and these are the cells which have been drawn here. And the likelihood is in an exam you'll be given an image like this and you'll be asked to draw some adjacent cells. Now we tend to draw some key sort of points of biological drawings. We're trying to draw what we actually see because they don't look like diagrams, and we're trying to draw the key features and the clear organelles. So for instance, you can see if we look at this cell which has been drawn here, which is pretty similar to the same shape, but it's not exactly the same, and you're not expected to be a really good artist. But what you have seen is this person has drawn the cell membrane and then they've drawn the nucleus. Okay. Now there's other things inside this cell that you can't see clearly, so you only need to draw the things that are clearly visible inside of a cell. Now here's an image of some common misconceptions and some mistakes that people make, and I'll clarify in a minute. Now on the left hand side, this is an image of a student's biological drawing where they've shaded and sketched, so this is not what you should be doing. Now this particular example is a tissue drawing, so you're trying to draw entire tissues rather than individual cells. So this person has started drawing lots of little cells on the inside, and you can see they overlap, but it's not very clear. They've sketched the line on the outside. It's various different thickness. You've got a darker line and then a lighter line. You've got this labelling, these crossing over of labels. Is this line going to here or up there, etc. Whereas this on the right hand side is an example of a good biological drawing. So here are the rules of a biological drawing. You need to put a title on, you need to put the magnification, but the actual drawing itself needs to be, think of it as drawing a graph. You need to use over 50% of the paper. You need to be using white and lined paper, and you need to use a very sharp pencil. And the official guidance from OCR is you should have a pencil nail file sharpener. It should be really very, very sharp. The lines you should be drawing should be very, very smooth. You don't sketch. So what we don't want you to see is you doing this. We don't want to see this around the outside of the cell. We want to see clear, continuous lines. And you shouldn't end up, as I've done there, I'm using a mouse very poorly, with this separation of the cells. The two lines, if you're getting to, they should be joining up perfectly to the best you can. And you rub it out, you use a very light pencil, and then you correct it if you need to. The proportions should be correct, and we'll come to that in a second. And the labels should all be stacked along the side on top of each other is the best way to do it. And you shouldn't be crossing the labels over on top of each other. Okay, and they should be parallel to the top of the page. So if this is our page, they should be parallel to that top of that page. So an activity I want you to do now is, can you have a go at drawing this cell? Now you can see here that there's two overlaps. So do your best to kind of go across there, but have a go at drawing this cell now. Okay, so if you've had a go, I'm going to do a very quick version here. You can kind of see, again that's very poor, I've been doing it on my mouse, it should be better than this. The key point you should be drawing is the outline of the cell membrane, and you should be drawing the cell nucleus, and then you should really only have 3 labels, and they should be the nucleus, the cell membrane, and the cytoplasm, because they're the only organelles that are clearly visible from here. You should have your title, you should have the magnification. Now I know you don't know this now, but this is what you should end up with, something like this. Okay, so because we've only studied a very short part of the course, I found that the questions that should be closest to what you can answer at the minute. Now I know this is a scanning electron micrograph, which we'll come to in further lessons, but I want you to have a go at this question anyway. Okay, so pause the video and have a go. Okay, so the answer is A. You could have worked out the magnification itself. If you have this on your paper, you can measure the size of it and you can do it that way, but also you should be aware of the rough size of the numbers, and we'll get to electron microscopes later in a later lesson. Next question. Okay, and the answer is A. So a microscope with high resolution is you can distinguish structures that are very close together. So resolution, the ability to separate objects as separate entities. Okay, so that's going to end this lesson. Just some sources that I've used. I've used Caboodle and Doddle for some of the animations and questions, used Google Images, and the rest of it's from me. Thank you very much. Goodbye.